So now the goal of gene therapy, what is the major goal of gene therapy? We say we could replace or we could insert a normal gene into a non-specific location. Non-specific location means this is not going to be getting into a specific location within the genome. Now, a non-specific location within the genome to replace a non-functional gene. Now, there is a faulty gene due to which we have a genetic disease, and we want to insert a gene into a location that we really don't know, and we just let it fire and get it into some location, but we hope that it's not getting in between a gene and it's not disrupting a gene. This is one of the most common approach. And then what happens next is like the abnormal gene is always swapped with a normal gene through homologous recombination. Homologous recombination we have discussed many times, and this is uh, done through insertion elements at the end of the gene construct. You always have a promoter and you always have the gene, a copy of your gene of interest, and at the ends of the flanking regions of the gene you always have these insertion sequences which has similarity, sequence similarity, or in other words you can also say homology to specific parts of the genome which help it to guide it towards that region of the DNA and integrate it into its position. Another approach is through selective mutation. So selective reverse mutation means if you have a point mutation in a gene, you could reverse the point mutation by another point mutation. So this kind of mutation is known as selective reverse mutation. You have one point mutation of, let's suppose, adenine to guanine, and then with another point mutation, you can change it back from guanine to adenine, and this returns the gene to its normal function. And of course, the regulation is very important. The degree to which a gene can be turned on or off. This is very much important in case of, let's suppose, certain cancer genes. When the genes are out of the regulatory machineries, you need to have regulation on them. When they go out of control, you could transfer a gene that could put the regulation, the gene expression regulation, back in control. Now, why do we get these genes are medicines? Genes are medicine because gene therapy is the use of genes as a medicine. For example, one of the good example in this case is hemophilia. Hemophilia is a disease in which when a person has a cut, the blood flow, keeps on, he or she keeps on losing the blood, and the repair of the fibrinogen is not as good as a normal person. It takes really long. The reason is there are two types of hemophilia, hemophilia A and hemophilia B, and in both cases, out of the 12 clotting factors, one of the clotting factors is missing. And one of the clotting factor is missing because the gene for one of the clotting factors is defective. In that case, what do? You inject that particular growth clotting factor. For example, factor 9 is one of the clotting factors. I think it's associated with hemophilia B, if I'm not wrong. Either A or B, I'm not confirmed, but factor 9. And what do you do in normal cases? You have to inject factor 9. So you have to purify, you have to clone the gene of factor 9 in yeast or E. coli, and then you have to purify it. And then you inject it directly into the blood. Now, that's the medicine. But if you go and do gene therapy and repair the gene of hemophilia, then you do not need to inject factor 9 into the blood flow of the patients, hemophilic patients. In this case, we say the genes are like medicine, because these genes, when they are inserted and incorporated into the genome of hemophilic patients, it acts like medicine for them. And as discussed a number of times already, the gene therapy actually delivers proteins because the end product of a gene after successful integration into the genome is protein. And these proteins are actually small biological factories. We could call them small biological factories, or the whole human body is not a small, but is a biological factory that produces the therapeutic protein. Like you have the therapeutic gene, that's the gene we call it transgene because it has to move from one place to the other. This is not its exact position, so like transgene. And then you have an efficient vector. This is something we're going to discuss in the next slides. You always need a vector. There could be, I mean, you could transfer it without vectors as well, but the most efficient way is to do with a vector, as we discussed in the production of transgenic animals. And inside the vector, that's the transgene, the yellow color. And when it reaches its destination, incorporates into a genome, it will be expressed, transcribed, translated. In between, there will be more steps. And eventually the protein, therapeutic protein will produce, and these therapeutic protein, it will cause its effect. Similarly, you can use vectors that are not, normally we use viral vectors, but you can also use other non-viral vectors. For example, this is a cationic liposome molecule. Inside the liposome, you have the plasmid and the CFTR gene, and this would go through the cytosis into the cell membrane in an endosome, and eventually this plasmid enters into the nucleus, and inside the nucleus there will be this CFTR is going to be transcribed into a messenger RNA, and it will be translated outside the nucleus as happens in eukaryotes. And of course you would have the protein as a result of this transgene incorporation, and this protein would act as a medicine, and the defective gene function will be restored through gene therapy. Now, with respect to cells, we can divide gene therapy into two types. Number one is somatic gene therapy, and the second one is germline gene therapy. The process is very much the same in both of the cases, but the difference is when we do gene therapy in the germline, what will happen? You will have a new trait, like as we discussed in production of transgenic animals. This is a new gene, and through micro injection, it has been injected into the blastocyst stage. We have discussed this, and then it will be implanted into a surrogate mother, and you would see the new trait. And you could also check for it by breeding if the gene is incorporated into the germline or not. When we have the gene in the germline, this means this particular animal in which we have done gene therapy, it could transfer it to its offsprings. In some cases, this is important, but in some cases, this is not necessary, and even in some cases, this is not needed, or it could be harmful. For example, there are certain genetic diseases which are caused because of exposure, or because of sedentary lifestyle, or because of environmental conditions. Now, if that is the case, it is not necessary that the offsprings will have this disease, because that's because of the exposure to mutagens. In that case, you do not need to do the gene therapy inside the germline cells. You want this transgene therapy to be confined only in the individual, and Unless he's alive, he should have these genes, but he should not be able to, he or she should not be able to transfer it. And in this case, you do it in the somatic gene therapy. So for example, you take any gene. Let's suppose you take the gene for insulin production, diabetes uh, mellitus 1 or 2. Now, if a diabetes, if a person is a patient of diabetes, it is not necessary that the young ones he or she would be having must also have diabetes. Well, he or she would be more prone to have it at a certain age or after a certain age. But during the birth, the new ones would be perfectly fine. In this case, we do not want the gene therapy to be in the somatic, we do not want the gene therapy to be in the germline, because we know that when the young ones are formed, this disease will not be in the young ones. But in case of, so that there, this is the place where we would do somatic gene therapy. But there are some diseases. If a father has this disease or a mother has this disease, the children are really at risk. In that case, you can do the germline therapy. For example, anemia, or not anemia, how do you say, thalassemia, sorry, beta thalassemia, alpha thalassemia. Now, these are the diseases, if you want to cure through gene therapy, you want to go the gene not only in the somatic, but also in the germline, because this could be transferred to the new offsprings.